Hello dear friends, today we are going to talk about the nucleic acids. I have taken this topic from the chapter of biomolecules and we are going to learn what nucleic acids are. My dear friends, we know nucleic acids are of two types. We have the DNA and we have the RNA. DNA and RNA. DNA means deoxyribose nucleic acid and RNA means ribose nucleic acid. In majority of the species, DNA acts as a genetic material, but in some viruses, RNA acts as a genetic material. But remember, DNA is the stable form of genetic material. RNA is not. Why I mentioned this, that the DNA is the stable form and RNA is not. Well, we'll dig inside and we'll try to learn in a quick while that the DNA contains the pentose sugar, and that pentose sugar lacks 2' hydroxyl group. And due to the lack of 2' OH group, DNA becomes a stable genetic material. Whereas in RNA, when we talk about the pentose sugar, that has a 2' hydroxyl group. And the 2' hydroxyl group, when the proton gets dissociated from the O, it becomes the 2O negative that acts as a free radical and that can form the internal bonds within the RNA molecule, therefore makes the RNA unstable. Although we'll try to learn that diagrammatically, but yes, for a while, you know that DNA is a stable genetic material because of the lack of 2' OH in the pentose sugar, and RNA is unstable because of the presence of 2' OH. Now, my dear friends, nucleic acids are the polymers of nucleotides. I will write this down. Nucleic acids are polymers of nucleotides. It means nucleotides are acting as a monomer unit for a nucleic acid. So in order for us to know what exactly the nucleic acids are, we must know what a nucleotide is. Why? Because these nucleic acids, these are the polymers of nucleotides. So if we understand what the nucleotide is, then what we have to do is we have to join these nucleotides with the phosphodiester bonds to make the nucleic acids. So now we will try to learn what a nucleotide is. My dear friends, you have to remember a single nucleotide contains 3 components. A single nucleotide contains 3 components. So first, nucleotide has 3 components. First is the nitrogenous base. Nitrogenous base. Second is the pentose sugar. The second is pentose sugar. And the third is phosphoric acid, or what we call as phosphate group. So phosphoric acid, H3PO4. It's also called as phosphate group. Okay. So a single nucleotide, friends, contains 3 components. One is the nitrogenous base, one is the pentose sugar, and one is the phosphoric acid. These 3 components togetherly form a single nucleotide. Now, what are nitrogenous bases? So first of all, we'll try to know nitrogenous bases. Then we will dig in the pentose sugar and the phosphoric acid. So nitrogenous bases. Nitrogenous bases. Nitrogenous bases are heterocyclic ring structures. These are heterocyclic ring structures. What? So nitrogenous bases are heterocyclic ring structures. Heterocyclic means that this nitrogenous base will contain 2 different types of cyclic rings, as we will see in the following diagram. So nitrogenous bases are heterocyclic ring structures. And under nitrogenous bases, we study purines and pyrimidines. So first of all, we will study what purines are. My dear friends, you will see that the purines are double ring structures. These are double ring structures. I have pre-drawn the diagrams in order to save the time. So Purines are double ring structures. Double ring structures. As you can see, my dear friends, this is the one ring, this is the second ring. And this ring, this is the sixth membered ring. We can see 1, 2, 3, 4, 5, 6. And another ring is 1, 2, 3, 4, 5. So purines are double ring structures. One ring is six membered and the second ring is five membered. So this is a six membered ring. Six membered ring, and this is the five membered ring. Although this is called as diazine ring, six membered ring is called as diazine ring, and the five membered ring is called as imidazole ring. These are not important terminologies in the biomolecules, but so that's why I'm not trying to focus on them, but you just know that one ring is six membered and the other ring is five membered. Now, when we talk about the nomenclature of the purines, you will have to start from here. This is position number 1. This is position number 2. This is position number 3, 4, 5, 6, 7, 8, and 9. So if you see in the six membered ring, we are going anti-clockwise. 1, 2, 3, 4, 5, 6, like this. We are going this way. So six membered ring has anti-clockwise direction nomenclature. So we go from position number 1, then 2, then 3, then 4, then 5, then 6. So this is the anti-clockwise direction. And the nomenclature of the diazine ring, five membered ring, is clockwise. Why? Because this was the sixth position. Now this 7, 8, 9. See, it goes like this: 7, 8, 9. 7, 8, 9. So this has the clockwise nomenclature. In the purines, the nitrogens are present. The nitrogen, it is present at the position number 1. At the position number 1, here is present the nitrogen. Position number 1, position number 3, position number 7, and position number 9. So, in purines, the nitrogen is present at the position number 1, 3, 7, 9. The rest of the positions contain carbon. Okay, so you have to remember the nitrogen positions because these are important. When we will try to study the glycosidic bond, that time you will see the importance of the position of the nitrogen. So, what are purines? Double ring structures. They have one ring six membered, one ring five membered. Six membered ring is called as diazine ring, and the five membered ring is called as imidazole ring. And the position of nitrogen lies at first position, third position, seventh position, ninth position. At 4 locations you will find the nitrogen. Now, under purines, we generally study two categories, two components. One is adenine, one is guanine. So adenine and guanine are the most important purines, which we normally study under the literature. So, now what is adenine? See, if at the sixth position, you attach amine group, if we attach amine group, if at the sixth position we attach amine group, it becomes adenine. So 6 amino purine. So this is a purine, and we added amine group at the sixth position, so it becomes 6 amino purine. And 6 amino purine is called adenine. So this is adenine. This is adenine. 6 amino purine. So what is adenine? 6 amino purine. Now what's guanine? What you will do? At position number 6, you will see an oxy group, and at position number 2, you will see an amine group. Amine group. So this is the position number 1, this is the position number 1, 2, 3, 4, 5, 6, 7, 8, and 9. So, what is guanine? See, 2 amino, 6 oxopurine. At the position number 2, we have the amine group, and at the position number 6, we have the oxo group. It becomes 2 amino, 6 oxopurine. So, guanine is 2 amino, 6 oxopurine. So, this structure is guanine. That is 2 amino, 6 oxopurine. So my dear friends, under purines you have to remember only these two structures. You have the adenine, you have the guanine. Adenine means 6 amino purine and the guanine means 2 amino 6 oxopurine. Double ring structures. Now, what are pyrimidines? Pyrimidines are single ring structure. Pyrimidines are single ring structure. See, we have this pyrimidine, and this pyrimidine. We can see this has got a single ring structure, single ring structure, single ring structure. And under pyrimidines, we study 3 structures. See, and we have to remember, in pyrimidines, the numbering, the direction of the numbering is always clockwise. We have, this is the position number 1, this is position number 2, this is 3, this is 4, this is 5, and this is 6. This is the numbering in the pyrimidines. It's different from the purines. 1, 2, 3, 4, 5, 6. Now, if you have at the position number 2 and the position number 4, the oxo group. If oxo group is present at the position number 4 and the position number 2, it is uracil. It is uracil. So I normally make a mnemonic called as 240. But this is not 0, this is O. And this is position number 2, this is position number 4. So O is present at the position number 2 and the position number 4. So the oxo group being present at position number 2 and position number 4. So what could be the possible nomenclature? 2,4-dioxypyrimidine. 2,4-dioxypyrimidine. So 2,4-dioxypyrimidine is called as uracil. So this is uracil. 2,4-dioxypyrimidine. Although this is not important to exact to remember the nomenclature, but yeah, in the NCERT, 2 structures are given. So you never know where from the question exact comes. Now this. See, we have the similar position. This is the position number 1, 2, 3, 4, and 5, and 6. At position number 2, we have the same oxo group as was in the uracil. At position number 4, we have the same oxo group as was in uracil. So it's a kind of uracil-like structure. But there's a difference. At position number 5, there's a methyl group attached. There is a methyl group attached. So, what I can make the nomenclature? Until this structure, at the position number 4 and 2, it seems like it's uracil, but at the 5th position, I am modifying it. I am attaching the methyl group. So, this structure becomes thiomethyl uracil. Thiomethyl uracil. So, thiomethyl uracil. And thiomethyl uracil is called as thymine. Thiomethyl uracil is called as thymine. Now this structure. At position number 2, this is 1, 2, 3, 4, 5, and 6. At position number 2, we have the oxo group, and at the position number 4, we have the amine group. So, 2-oxy, 4-amino pyrimidine is called as cytosine. 2-oxy, 4-amino pyrimidine is called as cytosine. So, this is cytosine. So, we have the 3 types of pyrimidines. We have the uracil, we have the thymine, we have the cytosine. So, all these structures contain single ring. That's why I mentioned pyrimidines are single ring structures. So we have uracil, thymine, cytosine under pyrimidines, and we have the adenine and guanine under purines. I hope we are now done with the nitrogenous bases. Now we'll take the pentose sugar. I will take the pentose sugar and we'll try to learn what exact pentose sugar is and what is its structure. Now, pentose sugar. we have two types of pentose sugars, 5-membered ring. In DNA, we have the deoxyribose sugar, and in RNA, we have the ribose sugar. So we have two types of pentose sugars. One is deoxyribose sugar, and the second is ribose. Now first we'll try to learn the ribose, and then we'll talk about the deoxyribose. Now ribose is a pentose sugar. Now how it is drawn? Like this. So this is the pentose sugar. 1, 2, 3, 4, 5. 5-membered ring. Now we'll try to learn the nomenclature. This is called as carbon 1, but it's called as carbon 1 prime. This is called as carbon 2, but carbon 2 prime. This is carbon 3 prime. This is carbon 4 prime, and this is carbon 5 prime. Now why prime? Why don't we use the normal carbon? Because we know in nitrogenous bases, we use the term carbon. Like position number 1, 2, carbon 1, like carbon 2, carbon 5, carbon 8, carbon 9, like that. So in order to avoid the confusion with the nitrogenous bases, we put a different nomenclature. We use the prime. We use the prime, carbon 1 prime, carbon 2 prime. So whenever in the DNA, whenever you talk about the carbon 1 prime, carbon 3 prime, so you must know you are talking about the pentose sugar. If you talk about simple carbon, then you must know you are talking about the nitrogenous base. So in order to avoid the confusion, we are making a different nomenclature using the prime word. Now, we see, at the carbon number 2, we have the hydroxyl group, and the carbon number 3, we have the hydroxyl group. So, in RNA, carbon number 2, that's carbon 2 prime, it contains the hydroxyl group. So this is, as I mentioned earlier, that this is what makes the RNA unstable genetic material. Because RNA contains this sugar, and this sugar has hydroxyl group on the carbon number 2 prime, and this hydroxyl group, upon releasing the H, it becomes the O negative, it becomes a free radical, and free radicals can make the internal bonds within the RNA, therefore making the RNA the unstable genetic material. Now, what is the difference between the deoxyribose now? Now, we'll try to draw the deoxyribose. So there is not a big difference, a little minute difference, but has a very huge importance. So this is OH, this is H, this is OH, this is CH2OH. Now what is the difference? Similarly, carbon number 1 prime, C2 prime, C3 prime, C4 prime, and C5 prime. So this is C2 prime, this is C3 prime. In the ribose sugar, which is present in the RNA, at carbon number 2 prime, we have the hydroxyl group, and in the deoxyribose, at carbon number 2 prime, we have only hydrogen. So this is the only difference. The rest of the structure you can see, it is 100% similar. So only the difference lies at the carbon number 2 prime. In the RNA, pentose sugar contains the hydroxyl group. In the DNA, pentose sugar contains hydrogen. Now why it makes the DNA as a stable and the RNA as unstable? As I mentioned, hydroxyl group makes the RNA as an unstable genetic material, but since there is no hydroxyl group present at the carbon number 2 prime, so there is no whatsoever case of making internal bonds within the genetic material. So this is the concept. Now you might be worried about, sir, there is also the hydroxyl group at the carbon number 3 prime. There is also the hydroxyl group at the carbon number 3 prime. Now, then this hydroxyl group can also make the internal bonds within the genetic material. Then how is it making the DNA stable and RNA as unstable? As we will see in a moment, 3 prime hydroxyl group is always involved in the formation of phosphodiester bond. So 3 prime hydroxyl group will never be free. It will be involved in making the phosphodiester bond. So I hope until this you are understood with the concept of deoxyribose sugar and the ribose sugar. Now phosphoric acid. Phosphoric acid generally, when we study the replication, the nucleotides come in the form of triphosphates, nucleoside triphosphates. 3 types of phosphates are there: alpha, beta, gamma. Now we will try to make a nucleotide. First we will make a nucleoside, then make a nucleotide, and then those nucleotides are joined together by the phosphodiester bonds to make the nucleic acid. Now we'll make a nucleoside, then we'll try to make a nucleotide. For example, now I will not draw the structure of nitrogenous base. For example, this is adenine. This is adenine. Now we know the structure of adenine. This is adenine. Or I must draw so that you will exactly understand how glycosidic bond is formed. So this is NH2 group, and there is nitrogen present here. There is nitrogen present at this position, position number 1. This is position number 3. This is position number 7, and this is position number 9. It has hydrogen as well. So this is position number 9. This is the adenine. Now I will try to make a pentose sugar. So the carbon 1 prime of the pentose sugar, although this is a ribose sugar, carbon 1 prime of this is the carbon 1 prime, this is carbon 1 prime, the carbon 1 prime of the pentose sugar, and position number 9 of the purine, because adenine is a purine. Again, carbon 1 prime of the pentose sugar and the position number 9 of the purine makes the glycosidic bond. See, H from here and OH from here. So H from here and OH from here, it makes a water molecule. So water molecule is removed off, and there is a formation of bond with position number 9 and the position number carbon 1 prime. So I can show it like this. So water molecule will be released. Here H and here OH will be released, and we can make a bond like this. And in this bond, water molecule is released. So always carbon 1 prime of sugar is used, and if it is purine, then position number 9 is involved in the formation of glycosidic bond. So this bond is called as glycosidic bond. This bond is called as glycosidic bond. So remember, in purines, carbon 1 prime, in purines, position number 9, and from sugar, always carbon 1 prime is involved in the formation of glycosidic bond. But had it been the pyrimidine, either thymine, either uracil or cytosine, then position number 1 of the pyrimidine is involved in the glycosidic bond with the carbon 1 prime of the sugar. If it's pyrimidine, then position number 1. If it is purine, then position number 9. And from the sugar, always carbon 1 prime is involved in the formation of glycosidic bond. So this is a nucleoside. Nitrogenous base plus pentose sugar makes nucleoside. Now what makes nucleotide? When nucleoside is attached with the phosphate group, it becomes the nucleotide. Since it was an adenine, for example, I mentioned it was adenine. Now this is the adenosine. When purine is, if we talk about the nucleoside of purines, we know purines have 2 structures, adenine and guanine. When adenine is attached with the pentose sugar, it's called as adenosine. If guanine is attached with the pentose sugar, it's called as guanosine. Sin suffix is used in the terminology of purine nucleosides. Now what about if pyrimidines make the nucleoside? For example, uracil is attached with the pentose sugar. It's called as uridine. Thymine is attached with the pentose sugar, thymidine. Cytosine is attached with the pentose sugar, thymidine. So din is used in the pyrimidine nucleosides. Okay, so this structure until this is called as cytoadenosine, because where the adenine, we have the pentose sugar. Since this is a nucleoside of purine, which type of purine? Adenine. So this structure is called as adenosine. This is called as adenosine. Now, we will attach the phosphate group. Remember, phosphate group is always attached at the carbon 5 prime. This is C5 prime. At the carbon number 5 prime of the pentose sugar, we attach the phosphate group. How? Like this. If we attach one phosphate, it's like this. So, when one phosphate is attached with the pentose sugar, what was the structure? Adenosine. And we added one phosphate. This phosphate is called as alpha phosphate. It's called as adenosine monophosphate. Remember, when we talk of the adenosine monophosphate, the bond present between the alpha phosphate and the pentose sugar, it's called as phosphoester bond. So until this, it is adenosine monophosphate. This is AMP, adenosine monophosphate. What about if we attach the second phosphate group? This was the alpha being attached. Now, this is the beta phosphate. Now we attached beta phosphate, 2-phosphate. Alpha is already attached, it was adenosine monophosphate. Now we attached the beta phosphate. It becomes adenosine diphosphate. And remember, the bond between alpha and beta phosphate, this bond, it's a phosphoanhydride bond. It's energy rich bond. Phosphoester bond is not energy rich bond. Now what if we attach the third phosphate? Like this. So this is the gamma phosphate. And the bond between gamma and beta phosphate is again phosphoanhydride bond, because it is the bond between the two acids, phosphoanhydride bond. So beta and gamma phosphates, these are energy rich phosphates because they have the phosphoanhydride bonds. And these bonds are actually used when we study the replication process. These bonds are broken down in the form of beta and gamma phosphate, and these release energy that drive the reaction forward in the replication process. So, it was first of all, it was AMP, adenosine monophosphate. Then we attached the beta phosphate. It became ADP, adenosine diphosphate. Then we attached the gamma phosphate. It becomes ATP, adenosine triphosphate. Okay? And we know that ATP is a complete nucleotide. And it is used as the energy currency of the cell. Remember, it was adenine. What about if it was guanine? Then it would have, first of all, it would have been guanosine, then GMP, then GDP, then GTP. What about U? If it was a uracil pyrimidine, then uridine first of all, then UMP, then UDP, then UTP. What about thymine? So TMP, TDP, TTP. What about cytosine? CMP, CDP, CTP. Although we can make CTP, we can make UTP, we can make TTP, or we can make GTP. Remember, among all of these, ATP is used as the energy currency of the cell, although the rest of the nucleotides can also be used in the as the energy currency, as we see, like glycogen biosynthesis, or we see the fatty acid biosynthesis. We use those types of energy currencies, but normally we use ATP. Why? We use ATP for the reason that we have this direct access for the production of ATP. We know the inner mitochondrial membrane contains that F0FN particle that is helpful in formation of the ATP molecules. So we have the direct access in formation for the formation of the ATP molecules. That's why normally we use ATP as the energy currency of the cell. Although rest like GTP, like UTP, those also can be used as the energy currency of the cell. So this is a complete nucleotide. Now, then this complete one nucleotide, it will be attached with the second nucleotide, then the second will be attached with the third nucleotide, and the process goes on. We make the polynucleotide. And that polynucleotide could be DNA, that polynucleotide could be RNA. So when we study the formation of the polynucleotides, the 3' hydroxyl group of the pentose sugar of the first nucleotide. 3' hydroxyl group, since the video has already gone long, so I will not explain that, but just a little concept. That the 3' hydroxyl group of the pentose sugar, it always will attack on the 5' phosphate of the incoming nucleotide to make the phosphodiester bond. Then the second nucleotide, which will be having the 3' hydroxyl group of the pentose sugar 3, that will attack the third upcoming nucleotide at the 5' phosphate. And the process will go on. We will keep on making the phosphodiester bonds to make the polynucleotide, and that polynucleotide could either be DNA or it could be RNA. As I mentioned, DNA will be the stable genetic material and RNA will be the unstable genetic material due to the presence of 3' hydroxyl group at the 3' carbon of the pentose sugar. I hope this is clear to you and made sense. Thank you.